Hello, welcome to our STEM News broadcast for week 6 of winter quarter in 2022. We are STEM News Nexus at UCLA, blog of preprints and new publications. For any accessibility related inquiries, please contact us. Our email is stemnewsnexus@g.ucla.edu. Check our description to sign up for our newsletter. Our website, www.stemnewsnexus.com, will be posted soon, and our references will be available in the description for your convenience. So let's get started with some STEM news.
Hi, everyone. Today, I am going to be talking about the COVID-19 virus. Specifically, I'm going to be talking about how genetic and immunological determinants affect the severity of COVID-19, leading to pneumonia. So as we know, the virus is benign in most individuals, but in 10% of cases, it does trigger pneumonia. So that begs the question, in what ways do genetics and age factor into the severity and illness of COVID-19? So there are several errors that are listed here on this PowerPoint that you can read about, and these are the molecular and cellular determinants that are critical in the development of the pneumonia that is associated with a severe COVID-19 illness. Many patients with these determinants may be unable to prevent the spread of the virus to their lungs, blood, and other organs during the first few days of infection, which leads to a lot of inflammation in the body. So that concludes with the fact that individuals carrying the at-risk genotype have greater risks and are more susceptible to stronger cases of COVID, especially if they are also elderly.
There is a lot more information on fat cellular biology. This review posted in Cell on February 3rd of 2022, entitled Adipose Tissue Plasticity in Health and Disease by Sackers et al., these researchers noticed how fat tissue accumulation is associated with disease and how it's known to have roles in mechanical cushioning and roles in both insulin uptake and energy homeostasis. Its cellular biology is being investigated to understand how fat cells bring about these physiological processes. So these researchers asked, what is the current understanding on how fat cells and tissues work together to respond to physiological cues? And there was a plethora of information, and I decided to focus on 4 that really interested me. So the first that I really appreciated was the discovery of PRX1 as a marker for subcutaneous fat progenitors. And this is the fat that's right beneath the skin, because fat development has been not as well understood recently until this point. And also that they noticed how fibroblasts that express W22 and PDGRFA will differentiate into visceral fat. So you can imagine all this extracellular tissue containing these fibroblasts, but at some point something's going to make them differentiate into the fat that actually coats the organ. And also they saw there has actually been some new lineage tracing experiments that identified fats to be originating from somite tissue. So it just seems like really interesting just to see whether they're actually coming from mesenchymal stem. In the past, it was thought that the fat came from mesenchyme, but now it's showing that they're coming from fibroblasts, which is pretty interesting. Something else that I really appreciated is how they noticed how sex and age influence the state of the fat tissue, which is actually a very dynamic thing, as we'll discuss in a moment. But essentially, hormones in the body are going to influence where the fat deposits will accumulate, and that this can be controlled experimentally by removing and administering hormones. Also, they noticed that actually there's hyperplasia and hypertrophy with adipose tissue. So hyperplasia is when there's a lot of cells. Hyperplasia is also a term used in tumors, like when there's excessive cell growth, hyperplasia, or plasticity or stuff. And then there's also hypertrophy, which is like the actual size of the cell increases. And what they noticed is that typically children, as they also have their hormonal things regulating where their fat is deposits are forming, that those eventually have a certain amount of members in their population at a particular age. And then what's causing the disease is that they increase in size. However, because with this next information, the adipose tissue is in constant turnover and changing, that adults can actually increase the amount of fat cells that they have because of this property. So essentially, fat cells are constantly dying and renewing, just like our skin. And they are also that macrophages, so once an adipocyte dies or a fat cell dies, a macrophage will eat it. And there has been some evidence showing that there's an axis of a secretory pathway that recruits progenitors to the area where there was cell death. So it seems like this kind of like, if you could just imagine just kind of like this actively regenerating, replenishing tissue, potentiates a lot of changes in the heterogeneity of the tissue and also just the population size. And also what they noticed is that aging in adipogenesis, or aging in general, reduces adipogenesis, or just like the development of more progenitor cells. So I guess that turnover is getting slower over time, and that there's also the induction or recruit and like active recruitment of these age-dependent regulatory cells that have the function of suppressing adipocyte progenitor cells to as a causal mechanism for how adipogenesis is reducing in age, in older tissue. And there's just so much more in this review. I highly recommend you read it. Ultimately, from everything that they gathered, they concluded that we as a scientific community have way more information that enables us to find new high impact molecular targets. The reason why I say high impact is because as mentioned, excessive fat tissue accumulation is associated with disease, but these are diseases that are very, very common in our population, like heart disease and obesity, that we definitely ought to improve our healthcare for. So just all of this new scientific findings is really potentiating some like really growth in a lot of growth in the treatment, management, and prevention of obesity mediated comorbidity stuff of like diabetes and heart disease. So yeah, okay, great.
So I'm going to be talking about how stress in the intestine influences chromosome inheritance. So the University of Cologne has discovered that signals from intestinal cells have a substantial impact on whether damaged eggs in the worm C. elegans are removed or not. So according to the study research team, they found that responses to environmental stress in the stomach now result in the production of a messenger molecule. And this messenger molecule controls the animal germline. So if control by the stress response in the gut is absent, the egg quality control fails. So they also demonstrated how stress reactions in the gut regulate the integrity of oocyte chromosomes, and the researchers understood how environmental variables impact oocyte quality through stress responses, and they give up whole new avenues for removing detrimental effects and preventing abnormalities.
Okay, welcome to our rapid fire section. So first things first, the Moderna just did the very first clinical trial of HIV vaccines starting on January 27th of 2022. And also there is a new electric patch that accelerates healing of dermal trauma or trauma in the skin. And we'll talk about this more next week. Have a good week, everyone.